Welcome to the BCHM 211 video on the levels of protein structure. This video introduces the fundamental concepts of protein structure. Proteins are vital macromolecules in biological systems. Their function is closely tied to their intricate three-dimensional structure. Understanding these structures is key to understanding biochemistry. Protein structure is often described in four hierarchical levels. These levels are primary, secondary, tertiary, and quaternary. The primary structure is the simplest level. It refers to the linear sequence of amino acids in a polypeptide chain. This sequence is determined by the genetic code. Each protein has a unique and precise primary structure. A change in even one amino acid can alter the protein's function. The secondary structure involves localized folding patterns. These patterns are primarily stabilized by hydrogen bonds. The most common secondary structures are alpha helices and beta sheets. Alpha helices are spiral structures. Beta sheets are pleated structures formed by hydrogen-bonded strands. The tertiary structure is the overall three-dimensional shape of a single polypeptide chain. It results from interactions between the R groups of the amino acids. These interactions include hydrophobic interactions, ionic bonds, and disulfide bridges. The folding is spontaneous and energetically favorable in most cases. Tertiary structure defines the active site in enzymes. The quaternary structure exists only in proteins with multiple polypeptide chains. These chains are called subunits. The quaternary structure describes the arrangement of these subunits. Interactions holding subunits together are similar to those in tertiary structure. Hemoglobin, with its 4 subunits, is a classic example. All levels of structure are crucial for protein function. Denaturation is the process where a protein loses its structure. This loss of structure typically leads to a loss of function. Denaturation can be caused by changes in temperature or pH. The primary structure is generally unaffected by denaturation. The folding process is highly complex and sometimes requires chaperones. Chaperones are proteins that assist in the proper folding of other proteins. Misfolded proteins are associated with several diseases. Examples include Alzheimer's and Parkinson's disease. The study of protein structure is essential for drug design. Knowing the structure helps target specific molecular interactions. Techniques like X-ray crystallography are used to determine structures. NMR spectroscopy and cryo-electron microscopy are also vital tools. The complexity of protein structure allows for their diverse functions. Proteins act as enzymes, structural components, and signaling molecules. In summary, remember the 4 levels: primary, secondary, tertiary, and quaternary. Each level builds upon the previous one to achieve the final functional form. Thank you for watching this video on the levels of protein structure.